Greetings to you all and happy Saturday after an exam week. I hope you're getting some rest this weekend and maybe a little bit of time off. Remember that we didn't have a chance on Thursday in our pretty chill trivia game to get to all of the questions. So for our pre-party today, I wanted to take you through the rest of those. And this first one maybe is one that many of you have already experienced, and that is looking at the differences between protein A and protein B. Protein A, notice that it spans the entire phospholipid bilayer within this cytoplasmic membrane, and recognize that we would call it an integral membrane protein because it does span the membrane. Sometimes it's also termed a transmembrane protein, whereas protein B is termed a peripheral membrane protein. It's interesting to think about experimentally how we would isolate these. Protein A would likely require us to add detergents to emulsify the membrane, whereas protein B, if we just changed either pH or added salts, we could most likely pretty easily isolate that. It's worth noting that there also is another type of protein called a lipid tethered protein, which would just be connected in some way via some sort of linkage to the membrane, even though it's more cytosolic in general nature. Recognize additionally that we might have thought that those integral membrane proteins were involved in being transporters. And last time you had mentioned that as one of the answers to the question. When we think about what kinds of molecules can flit across the membrane freely without needing to have some sort of transporter, I like to think about an analogy. So if you've ever been to a pretty nice dance club, you'll remember that they tend to be pretty picky about the people who can come into the dance club. And so I like to joke that they let people in, that they let girls in that have low molecular weight, and those that don't want to drink water, hell no H2O. So we can say that by analogy, those molecules that are tiny, very small molecules, but also those that are hydrophobic, that are not polar in some way, so that they can flit across the nonpolar tails of the phospholipids. So that means that other things need to have some sort of transporter. And so there are a variety of categories of transporters that we can look at. The channels and pores are those that do not become saturated. So they essentially have a linear dependence upon concentration of whatever ligand they're transporting. Whereas, and they're passive, you know, they allow things to move down their concentration gradient. Now, those that are facilitated diffusion passive transporters, they're a little bit different because what they do is with each ligand that binds, they undergo some sort of conformational change that then allows that ligand to be brought inside. So because they are accommodating one molecule at a time, they are saturable. And that's different than channels and pores that essentially just allow free inflow of these ligands or these molecules that are polar or charged that they're allowing to come on in. In fact, we could go ahead and draw this. We would essentially see that linear dependence for channels and pores on the concentration of the solute that they're binding, versus when we're talking about the facilitated diffusion transporters that accommodate one substrate at a time. This is much more like an enzyme because it undergoes a conformational change that means that it can only accommodate one substrate molecule at a time. So we see this much more hyperbolic dependence. So this reminds us of something, and we're about to get there. But I just want to make note that the third type of transporter is one that does require energy. So channels and pores, facilitated diffusion carriers, these are all passive. Active transporters require some sort of energy input. Now, when we're differentiating between bacteria and animal cells, the pores are the term we use for the non-saturable transporters, whereas in animal cells we tend to call them channels. So you can see that, of course, they provide that nice central area for passage of hydrophilic molecules. And of course, the pore lining would have the hydrophilic amino acids to allow that transport to be favorable. So they diffuse down their concentration gradient, no requirement for energy. One very famous type of channel in human systems is potassium channels. And it's cool to think about the two amino acids that we would tend to see lining the channel in order to bind with and favor the transport of a cation like potassium. So of course, aspartate and glutamate. Some of you did well with those kinds of concepts on the exam. So aspartate and glutamate, or D and E one-letter abbreviations we would find lining that. It's really cool to think about potassium channels because they actually will transport potassium 100 times faster than they transport sodium. And that's because of the size of the ions. So remember that potassium is a much larger ion. And so the channel is set up to desolvate the potassium more adequately than the sodium, which is a smaller cation. And so that just in and of itself accounts for the fact that it tends to want to transport potassium over sodium cations. So we contrast those channels and pores with the facilitated diffusion transporters that are saturable, and recognize that the difference here is that conformational change that makes it requisite that the transporter accommodate only one ligand at a time, and therefore you can see how there would be a traffic jam. I like to make the analogy that these are like when a ship goes through a lock. There has to be one ship in the lock at a time as it's moved down to the lower level, and it can't accommodate more than one ship. So if there's more than one ship, it has to wait in line, and that's what's happening here as well. So we talked about what the function would look like for that, and that it would be hyperbolic, and then I think you got to this notion that it's probably also described by something that looks a lot like the Michaelis-Menten equation. One big difference about it is that instead of the Km, we see a K transport, which is analogous to the Michaelis constant, and it does still describe the concentration of substrate at which the transporter is half saturated, or we could say the solute, whatever ligand that it is transporting from the outside to the inside. So we can kind of think about the difference between the concentrations out and in. So we know that if we're talking about bringing something from the outside of the cell in, eventually we will reach this place where the concentrations inside and outside are even. Then of course the transport will no longer take place because there is no concentration gradient to be had, and that's what drives passive transport. So I want to talk just a bit about active transporters, but as we move towards that, I think a couple terms are requisite. First one is antiport, which is when you have a transporter that transports two things in opposite direction, and the other is symport, where there are two things that are transported in the exact same direction. The two types of active transport that are important for us to think about are primary and secondary active transport. In primary active transport, we see ATP being hydrolyzed as the source of energy. So this is a good example of this is ABC transporters, or ATP binding cassette transporters. And they do hydrolyze ATP in order to bring in a solute, but that solute is bound to a solute binding protein that helps to adapt it to transport by the integral membrane portion of the transporter. So it's really, really interesting to note that ABC transporters are used in very unique ways in bacterial cells and in tumor cells. Many of our multi-drug resistant bacteria actually have found ways to utilize their ABC transporters in reverse. That is, they found ways to essentially turn them around to pump drugs out of the cell. And that's one of the features that can give a bacterial cell resistance to antimicrobial drugs. Additionally, tumorigenic cells will also do this in order to pump out chemotherapeutics. So it's really interesting to note that ABC transporters are highly studied because of that, and rightly so, because if we can work through the resistances that are seen due to these ABC transporters, then treatment will be more accessible to us. The other type of transporter that's active is called the secondary active transporter. Secondary active transporter, instead of relying on ATP, it relies on the gradients. And the most famous of those gradients is the proton motive force, or PMF. So in this case, we see a lactose permease in the bacterium E. coli, where the proton binds first to the transporter, and then that powers the influx of lactose. So I like to think about this as sort of writing down a gradient, the way that you might, so if there's like dammed water and it's pent up above the, say, power plant, it can turn turbines that can convert it to electricity. This same notion is true here. You've got this sort of pent up proton motive force that can power certain things. And in this case, the lactose is essentially riding the stream of protons down the gradient. I have this fun analogy that if you've seen Finding Nemo, there's this one part where the turtles are riding the EAC, Eastern Australian Current. So in this case, the lactose is riding the PMF to get on into the cell. So secondary active transport using the gradients. The gradients are not just protons. We also see gradients in the form of sodium and potassium cation gradients, and the sodium-potassium ATPase is responsible for that gradient. Notice that this transporter is an antiport that pumps 3 cations of sodium out and 2 potassium in. So recognize that that leads to a net difference in the charge on the inside and the outside of the cell. There's a net negative on the inside. So this does require ATP, so it is a primary active transporter, but recognize that it does generate gradients of both sodium and potassium. This is absolutely essential to mammalian cells because it does have that, it does give that net difference in charge, but also this maintains osmotic pressure in such a way that cells don't burst. So solids compatible with the cell are used to do that. So the sodium-potassium ATPase is very, very famous for many reasons, or it's also called the sodium-potassium pump. But the other thing to note about it is that it does create this sodium gradient, and so then this sodium gradient can be used for transport. And here on the right, you see the sodium cation gradient being used to transport glucose. So that glucose is riding the sodium gradient.